Hello, my name is Theresa Wiessner-Kroh. I studied biochemistry and have a PhD in biology, and since 2021 I'm working for RGCC as a scientist in the cell and molecular biology, and since the beginning of this year I am the lead scientist for this area.
My name is Stefan Rubner. I'm the head of laboratory of our RGCC R&D facilities in Halle in Germany. And I'm almost since 6 years in RGCC, and I take care about all the lab management and lab development.
So our main goal is that we want to identify novel targets that we can address with anti-cancer drugs, and we also are interested in identifying novel biomarkers, and last but not least, studying basic cancer processes to understand this disease more and more.
Our research is special about that we can combine all the disciplines needed for basic and applied cancer research. We have laboratories caring about the bioscientific area together with our colleagues in the medicinal chemistry departments, developing new therapeutics that we can test. And then, of course, we have all the relations to the clinic, getting clinical samples, evaluate them, analyze them, and also we are in close contact with our clinicians who see it on the patients how our testings and our therapies are working.
I think that especially by identifying novel targets and biomarkers, we will improve more and more the diagnostic part, and with this then we can also improve more and more the individualized or personalized therapies.
For me, there are clearly 4 points that I want to address. First is by finding new biomarkers, our diagnostics of cancer will be improved to detect all the cancers as early as possible. Second is about the treatment. I guess we will have more chemistry and biology related approaches to even drug undruggable targets, which are considered as undruggable so far. The third point is that we will make advances in cellular therapies. And the fourth issue, or fourth point on which we are working really a lot is understanding the tumor microenvironment, all of its complexity. And all 4 points we can address in RGCC with our colleagues. And this is what it's really outstanding to work for RGCC.
For example, we are collaborating with the laboratory of Greece, and from this laboratory we get the RNA which is derived from the CTCs that we can analyze it further, and this data will be then introduced in our pre-drug workflow.
So to achieve our goals, I would say from my side we have like 2 big goals. First is the identification, validation of new cancer targets that we can drug, and also the identification of new biomarkers for diagnostic purposes. And then, of course, we want to evaluate all of kind of drugs to proceed them further for clinical testing. And here we have the great opportunity to get a lot of clinical data for our biomarker and target identification from our colleagues in RGCC from Greece. They provide us clinical samples, which is really valuable for our clinical relation of our cancer research. And second, with the chemistry department, the medicinal chemistry in Wäldenswil, they provide us a lot of potential drugs, small molecule inhibitors that we can test back in our lab for further evaluation.